Hi everyone, it's Michelle from Iglio. I'm going to be narrating the PowerPoint for the urinary system in your Marib textbook. And it's kind of a longer one, so if you want to stop halfway through and come back to it, that would be probably something that's smart. Um, the urinary system itself is not a super complicated topic. What's a little bit more conceptual is how the nephron works and how the hormones work that go with water balance and blood pressure. So just keep that in mind. A lot of the urinary system is structures, but again, there's a couple of topics that you'll probably be, it'll be really important to be looking at possibly some videos or animations of how this works. So I will guide you through that and we're going to get started. Okay, so the first thing here, metabolism requires oxygen and nutrients. So what metabolism is, it's defined as the sum of all of the chemical processes that happen within the body. So we're talking about all the things that are built up, all the things that are broken down, and all the waste products and how these things are manipulated in the body. So when we metabolize things, we produce waste products. And we've talked quite a bit about systems that excrete some of the waste. And hopefully you can figure out that the digestive system obviously is going to eliminate a lot of waste. We also do eliminate some waste through breathing. Carbon dioxide is considered a waste product. And then our integumentary system, our skin, also has a little bit of excretory function. But the urinary system, being a system that filters all of the blood in the body and fine-tunes that to decide what to eliminate and what to keep, the nephrons are really an essential homeostatic structure. Okay, so let's discuss the main functions of the kidney. So the first thing is that the kidneys are going to filter blood. They can filter 180 liters of blood a day. That's insane. Obviously, we don't have that many liters of blood in our body. We have, anywhere between 4 and 6 liters in general. The 180 liters means that we're just going to keep circulating the blood and filtering it and cleaning it and, balancing what's in our blood to keep us within our homeostatic range. And so the things that are going to leave the body are going to be toxins and poisons, waste from metabolizing, wastes from, medication and drug remnants, extra water, extra sugar, like whatever we have that's not within its balance, within its range, is going to be eliminated from the body somehow. That's what homeostasis is. So the first thing is that it's going to filter blood. Second thing that kind of goes with that is the volume of the volume of fluid in your body is going to be regulated by the kidneys, right? So the kidneys, if you have too much fluid in your body, the kidneys can eliminate some. If you have not enough fluid in the body, then the kidneys can conserve that fluid. And then blood pressure goes really, really closely hand in hand with fluid balance, but there's also a few other things that are released from the kidneys that are going to help with blood pressure. It's not necessarily involved with water, so we'll get to that. As far as ions that are in the blood, we're talking about smaller, really you just call them smaller chemicals. But they are, sometimes we have too many of these, or sometimes we have not enough of these, and the kidney will pick and choose what our body needs at that time and eliminate the things that could be possibly a problem. pH of blood, we discussed already that the respiratory system does a really awesome job at at helping to balance that pH, but it can't do it alone. The kidneys are going to do a good job at working with the respiratory system, I guess you could say, to help balance pH by maintaining the buffers in the blood, and produces hormones. So the first one is erythropoietin. We call it EPO. And that drug, well, that chemical, that hormone is going to be involved with signaling the bone marrow that we need to make more blood. So we'll talk about that. And we also have renin, and antidiuretic hormone is another one we'll talk about, but antidiuretic hormone and renin, they're complicated topics we'll get to at the end. So let's move forward. All right, so here are your structures. I am hoping that my picture doesn't block this for you. I'm not really sure how to move it, but let me give you a page number just in case. So this is on page 975 in your text, so you can see that cadaver one. All right, so the function of the kidneys. We talked about all the different functions, but one of the big ones is, you know, filtering the blood and getting rid of waste. Because if you have waste products building up in your blood literally becomes toxic. pH changes, cells start to die. Like you cannot live without this function. So when our body breaks down proteins, proteins are made up of little chains of amino acids. And when we break down proteins and get these amino acids, and when we use these amino acids, we can create some really toxic chemicals that need to be eliminated. Ammonia is one of those. Now, if you can picture ammonia in, you know, if you've never smelled ammonia, it's powerful. It will burn your mucous membranes. It is a dangerous chemical really to be using. We don't really clean our houses with ammonia all that much anymore. So ammonia is pretty nasty. The liver converts ammonia, and ultimately we end up with urea, which is going to be excreted from the body. So we really, you know, we have these poisonous things, and our body works together to eliminate them. Liver, respiratory system, all of these systems. So then we have creatinine. Creatine is actually creatine phosphate. That's a chemical in our body that is going to allow for a lot of energy. A lot of bodybuilders like take creatine to help with muscle mass. When that's metabolized, it becomes something called creatinine. That needs to be eliminated from the body. Uric acid, that kind of goes with our amino acids. I'm sorry, nope. Uric acid comes from nucleic acids. So our nucleic acids are things like DNA and RNA. And then also foreign substances. So I already kind of said this. This is a little bit more reiterated here, but we're talking about things that shouldn't be here in our body necessarily, things we're putting into our body that don't really naturally belong there. So these are the four main organs to the urinary system. First one is the kidneys. That's the most complex. That one we'll talk about the most. The kidneys are going to filter the blood. It says blood plasma here, but it's really the blood in general. So the water and solutes, which are things that are dissolved in the water, not all of them, but most of them will return back to the blood. A lot of them we'll talk about in a little while. And then the kidneys, you know, they pretty much concentrate this waste that they've sorted, and then we, you know, so they make urine, which we call filtrate for a while, but we'll get to that. The ureters are these little tubes that run straight down and go down to the bladder. And that's what they do. They transport urine. They use peristalsis a little bit to make that happen, which is the same thing that we saw in the digestive system. And then we have the urinary bladder, which is just a storage sac. It's just a little storage sac for urine, basically. And then the urethra, which is going to eliminate the urine to the wherever you're urinating. So out of all of these things, the kidney is really the one that really does anything substantial. The other ones are just bringing this stuff out. And so, I mean, technically, without a bladder or a urethra, like, you can survive. You just have to, they have to use a conduit, kind of like a colostomy bag. But the kidneys themselves are doing the work. All right, location-wise. The location of the, I'm just going to give you a couple page numbers again because I'm not sure if you can see this. So this picture and the picture below, I believe, are like 976, 977-ish. All right, so we learned all about retroperitoneum. We talked about this mainly when we started discussing the digestive system. Quarantine is and making it hard to think. I'm sure you're realizing this too. So I'm going to get my words out. So retroperitoneal means that the organs that are located retroperitoneally are organs that are sort of smushed to the back wall, closer to the back wall of your digestive system and excretory system. So the peritoneum is a membrane that surrounds the whole abdominal cavity, and then it forms sort of like a pocket that all these organs sit in, but some of the organs sit behind that pocket, and that is called retroperitoneal. So retro means back or behind. And so the kidneys and the ureters and part of the bladder all are retroperitoneal. I know we didn't get a chance to really get to dissecting things, but when you do dissect if you get to dissect, when you first open up a mammal, if you remove those organs that are in the front, most of the digestive organs, there's like, you can't even see the kidneys right away. You have to kind of like break through the peritoneum to get to the kidneys. So the location is retroperitoneal, and they are lateral to T12, L1, L2, L3. One is slightly lower than the other, and that's because of the liver. So the liver is sort of a wedge shape, and so it's larger in one side, and so your right kidney is going to be a little bit lower because that's the arrangement based on your other organs. All right, so average kidney is 11 centimeters by 6 centimeters by 3 centimeters. Everybody's is slightly different, but if you can picture a bar of soap, like, white soap, what are they, Irish Spring or Dove or any of those, that's about right. As far as size-wise, it varies again by person. So again, the right kidney is lower than the left. So we talked, when we did the respiratory system, we talked really, really briefly about the hilum. A hilum is a location where the blood vessels and nerves would enter or exit an organ. So we talked about the hilum of the lung being a location where the bronchial tubes and the pulmonary vessels would enter the lung. So in this case, we have the hilum of the kidney. This is where we have our renal artery, our renal vein, and our ureter going down. And looking at the kidney itself, the kidney has 3 layers, and from superficial to deep, okay, so from outer to further in, all the way on the outside of each kidney, we have a coating or a layer that's made of dense connective tissue, so it's very strong. It's called renal fascia. So the renal fascia is, it's tough, it's strong, and it helps anchor the kidney into place, so it's going to help kind of hold it where it's supposed to be. The second layer in, so just deep to that, is adipose. So it's fatty tissue. It helps pad those kidneys and also helps kind of wedge the kidneys in place. And then we have our fibrous capsule all the way on the inside, kind of touching the kidney itself. And that is made, again, of a connective tissue. That capsule makes kidney infection, it prevents kidney infection from spreading throughout the entire abdominal and retroperitoneal areas from mixing. So let me rewind and say that again. So the capsule itself is gonna, if there's an infection within the kidney or an infection in the cavity that the kidneys are in, it like keeps them separate. Is the best way I can think of it. So when an organ is encapsulated, that's what that means. It means it's like there's like an extra layer protecting it. The thyroid is similar. So thyroid cancer rarely spreads other places. It's kind of the same way. All right, so let's move on to, let me just get my notebook here. The next image is on it's the same as 976, but there's an extra one. So we have their 976 down here, or up here is that cavity I was discussing with you. So let me see if I can start doing some writing here. I have a new monitor, so my monitor is in front of me, and then for some reason, I don't know, I have to use my laptop at the same time in order to see myself and see my pictures. So if I'm looking up and down, that's why. Okay, so I'm going to use this black, and hopefully I can write. So peritoneum here. So we have two peritoneal layers. So the peritoneum that surrounds the whole cavity is called the parietal peritoneum, and then the peritoneum that sits on the organs would be called the visceral peritoneum. Well, what we're dealing with here is this layer is all peritoneum, and inside here would be your digestive viscera mostly and all that stuff. So the kidneys are these little blobs. These guys are sitting along the back wall, and here's your vertebrae, spinal column, right? So these are your vertebral bodies. Our body is this guy. So we have our kidneys here and here sitting behind your peritoneum, retroperitoneal. And then the bottom is just a, diagnostic imagery that's going to show the same thing. Okay, so this exact picture is on 977, and next to this picture there is a frontal section as well of a kidney, a dissected kidney. So these are the parts to know. We have renal cortex, renal medulla, renal pelvis. Those are big. So looking over here at your kidney, the cortex of the kidney is this outer zone. Cortex means bark. So if you're picturing a tree right now, the bark of the tree is called a cortex. So if you think of your brain, you have the cerebral cortex on the outside. Your renal medulla is the sort of whole middle zone, all of this. The medulla has some other structures within it. And the structures, let me erase this, are these little triangle shapes. Those are called the renal pyramids. And at the tip of these, there's a little sort of, this is very hard to do. Okay, there's a little sort of spot right here called the renal papilla. Pap means bumpy. And that is at the apex. So remember I taught you a while back that the apex of something isn't necessarily the top of it, it's the pointy part of it. And so with the heart, the apex is that pointy part at the bottom or at the inferior boundary. All right, so, and then the last thing is in between these pyramids, we have kind of a columnar shape. And so this is called a renal column. So we have, I'm going to use like a yellow because urine. All right, so what we have going on is we have blood. We're going to talk about this over and over. I'm going to repeat this stuff over and over, so don't stress if you don't get it right this second. So no, the yellow's not going to work. Let's do green pea. So the P, I can't find my little, oh, it's way over there. Sorry. This is not working out very well. Okay. So the blood is going to enter, where is my, it's not writing. Okay. So the blood is going to enter through the renal artery. That's what this is. So the renal artery is going to branch and branch and branch into smaller and smaller vessels. Eventually, and this is all kind of happening behind this, it's going to go out and reach this cortex. And in the cortex, we have it's actually a million different structures called nephrons. I lost my mouse again. Okay, so here's our nephrons, and the nephrons filter the blood, and the stuff that has to leave our bloodstream is going to go through these pyramids, and it's going to enter into this region, and then it's going to drain into this region, and then it's going to drain down the ureter. So I'm going to erase this. all of this junk that I did. Hold on. I'm going to try to do this more neat. I apologize. Again, technical difficulties, quarantine, all that stuff. Okay. Okay. So the area that I mentioned that's in here is called the renal pelvis. Now, so we separate our waste. So the good stuff, the stuff we want to keep, is going to kind of drain down blood vessels and exit this way through the renal vein and return to the bloodstream. The stuff we don't want is going to go down the ureter. And so our structures are going to be the urine flow is going to go down through our nephrons as we make it. It's going to collect sort of at this little cupish area right here. That's called a calyx. The minor calyx is little. And as it continues on, we have sort of a larger cup shape, which is our major calyx. And then it drains into our large region here called the renal pelvis. And then from there, it goes down our ureter and down to the bladder and down to the urethra, and then it will exit. No, and the urethra, yeah. All right, so here's just another diagram, same thing, same one that's on 977, but this time it's labeled for you. So we have our renal cortex, renal medulla, we have our major calyx, minor calyx, renal pelvis, ureter, pyramids and columns, and then our fibrous capsule. I'm going to pause this for a sec. Okay. All right, so I'm going to move to the next slide here. And this slide is going to be a little bit complicated because some of you have gotten some of you have a background in the autonomic nervous system and some of you don't. And unfortunately, I don't have the ability to do an entire lecture on the autonomic nervous system at this time because it's a really hefty topic. And again, things are a little messed up right now as far as classes and the virus and all of that. So I'm going to really give you a really quick, just sort of one slide on the nerve supply that goes to the kidney. And so there's a grouping of nerves that is called the renal plexus. So I know that you all learned about the spinal cord and the plexuses of the spinal cord. So we had like the cervical plexus, the brachial plexus. There are also other plexi or plexuses that go to different organs. And so the renal plexus is going to go to the kidney. So renal and nephro both mean kidney. And so the renal plexus is a network of autonomic fibers. Autonomic fibers go and involve the structures and functions that we don't have conscious control over. It branches off of a different plexus called the celiac plexus, which is located, if you think about celiac generally involves stomach. And so if you can think of your celiac trunk. So if you picture going just posteriorly, then that's sort of the area we're talking about. And so it's supplied by the sympathetic fibers from the lowest thoracic splenic nerve. So that's a whole different game. But remember your sympathetic nervous system and your parasympathetic nervous system. So I hope that you got a little bit of that. But your sympathetic nervous system or sympathetic branch of your nervous system involves your fight or flight response, which is going to have a huge impact on many aspects of your body. And the kidneys are, how do I explain it? There's also involved with the kidneys, there's also the suprarenal glands. So those glands are also known as your adrenal glands, which has a huge, huge role in the sympathetic nervous system. So your adrenal glands create adrenaline. Kidney function changes based on whether your body is having this sympathetic overwhelming response to something. or this fight or flight response to something. And so these nerves send feedback to the autonomic nervous system, and vice versa. I can't explain a whole lot more because your background knowledge of the autonomic nervous system is not gonna allow for us to go further into this topic right this second. I'm trying to think of the words that would work, but you kind of have to have that background knowledge first. So I'm gonna just have you, remember that the renal plexus, I was trying to clear this slide and start over, but I don't know how. Sorry. Okay, so the renal plexus is going to be an offshoot or kind of a branching of the celiac plexus. Let me get my mouse. Celiac plexus right here. And our renal plexus is going to branch off of that. And this guy was supplied by sympathetic fibers, and here's those other nerves that I was referring to. And that goes with lumbar nerves. So don't stress about this right now. I would understand that, it's not simple as far as nervous stimulation to the kidneys. So the biggest part of all of this is that the renal arterioles, which are the blood vessels in the kidney, the diameter of them is incredibly important because the amount of pressure that's going into the kidney or into each nephron really is super important, and we'll talk about that in a little bit. I'm sorry, this slide was kind of a mess, so we're going to move forward. All right, so your nephron is a functional unit of the kidney. We have about 1 million nephrons per kidney when you are born. They do die off as we get older, but they do also, the kidney also has a really awesome regenerative capability. I'm just trying to find this actual picture for you. It's kind of a little bit ahead, so 985. I mixed up the order of this just a tiny bit, and then also your book has like a description of something, and then the picture's like on the next page or the page after, so I wanted to kind of go in order from where it's filtered, forward through the pathway. And the best way to start out here is to start with the blood vessels that are actually bringing stuff into the kidney. So we're going to start by talking about that. So this whole area in here, there's a filtration thing going on. All right. Okay. So this whole structure that you're looking at is a filter. And what we have up here is a blood vessel called the afferent arteriole that is bringing blood into each individual nephron. So now this cup that we're looking at is part of a nephron. So the nerves I was talking about on the previous slide, involve maintaining diameter of some of these blood vessels. All right, and so blood is going to come in through the renal artery and do a lot of branches, and eventually enters in through the afferent arteriole and enters this structure. This whole structure is made of a bunch of different things, but it's called the renal corpuscle. And its job is to filter plasma. I'm just going to say filter blood. And so the blood comes in, and then it enters into this weird sort of capillary knot. So you can see all these weird little blood vessels going on in here. That whole knot is known as a capillary network, and we call that the glomerulus. The glomerulus. So we'll talk about the complicated process that happens there. But this is the glomerulus. Blood comes in and enters the glomerulus. And if you can see these sort of little slits that we have here, these act very much like a filter, and they separate things according to size. So to me, the best way to remember what happens here is that we are separating things, and the big things are going back out through this tube, the efferent arteriole, and the smaller things are made it through these little pores, and they're returning and they're moving this way. So we'll talk about all of this as we go. So one more time, I'm going to erase this. Okay. So this capsule that captures the stuff that was filtered, which we call filtrate, is this capsule is called the Bowman's capsule or glomerular capsule. And it's a double layered wall of, or cup of epithelial tissue, epithelial cells. And, you know, as we separate by size, the filtrate, the stuff that, that was small or dissolved in water, is going to continue this way. And the larger materials are going to lead through this blood vessel and eventually will return to the body. Not all of it, but some of it. So then, if you can't see this part right here, then you will be able to in a little bit. So then what happens is the stuff that still has to be filtered is going to proceed forward, and it's going to enter a section of the nephron called the proximal convoluted tubule. Proximal means closer to where it started. Distal means further away. So it's going to enter part of a tube called the proximal convoluted tubule, and it's going to loop around, and it's going to go down a little, they call it a hairpin or a bobby pin loop. It's called the loop of Henle, and then it's going to continue along through a little more of a tube called the distal convoluted tubule. And we're going to discuss what happens at each step. All right, to form urine, there are 3 steps. And if you're watching this as part of my 2202 class, 2202 virus class, I guess you could say, of 2020, we had 3 steps, and you had an assignment that took you kind of walked through, or maybe you haven't done it yet. These 3 steps, it was a web quest. So the first step is glomerular filtration. And this is where we are very simply dividing things by size. So the best way, I think there's 2 examples of ways that I picture this in my head. One is when you go to the beach, and you know those kids' toys that are like a sand sifter, and you put the sand in it and you kind of shake it, and the big stuff stays in there, the shells and the rocks and the fun stuff, and then the sand falls through because the sand was smaller and the rocks were bigger. That's what happens here. A more accurate representation, I think, is old school way of making coffee, not the pods, but an actual coffee filter, a little piece of paper, you put your coffee grounds in it, and you add water, and the water like picks up some of the coffee chemicals, and the water seeps down and forms our coffee. But what's left in here are the coffee grounds. So if you can picture the glomerulus being this coffee filter, and the filtrate, or the thin stuff, the little stuff that is dissolved in water, that moves down and enters into the rest of the nephron. And then the coffee grounds, we would throw them away, and your kidney doesn't throw them away right away. They decide, okay, what's in there that we might need? Just because it's big doesn't mean we have to throw it away. So a lot of that filtrate is going to be junk we throw away, but some of it isn't. And so that's what happens in the next few steps. So the first step, your blood pressure is going to force water and solutes, things dissolved in water. It's going to force those across the glomerular capillary. It's going to force them across the walls. So that's filtering. The second stage is called tubular reabsorption. And so as this filtrate moves forward, it's going to enter into the first part of the urinary system, and a lot of that we just discussed is water. And so we don't want to get rid of a lot of water. Like, we want to keep, remember, your plasma is 90% water. We don't want to get rid of all of that water. And so the tubes, the rest of the nephron, the tubes, they return that water, they reabsorb that water, they send that water back into the bloodstream. And also a lot of things that were dissolved in the water, we don't want to get rid of. Just because it was small doesn't mean we want to eliminate it. So first we filter by size, and then we're starting to fine tune. Okay, the little stuff that was dissolved in water, you know, we don't want to get rid of all the water. So let's okay, it was great to filter it, but let's put it back into the bloodstream because we can't get rid of everything. And so the way it does that is it moves through these very thin walls into capillaries, just like how the lungs did gas exchange. The renal tube is going to bring blood back into these peritubular capillaries. And then tubular secretion is kind of the last part of that, and that's where the large particles that were, they kind of stayed in the blood, they're not necessarily all garbage either. So you have to kind of fine tune that and bring some of that back into this renal tube. Ultimately, we are fine tuning, we're passing back and forth the things that we might want, we might not want. We're kind of making that decision. Our kidneys are deciding what needs to stay and what needs to go. This stuff that I'm explaining to you, I'm going to talk about for the next however many slides, I don't know. But the best way to picture what I'm telling you right now is to watch a video of the process, an animation of the process. Watch those crash course videos that I mentioned, the urinary system crash course. This stuff right here, it's like that sliding filament theory of muscle contraction. Like you have to watch a little animation of it for it to make sense. So we're going to move on and we're going to just talk about these structures or these functions a little bit at a time, and I'll walk you through it. So this is like a super simplified version of a nephron and what happens here. I know there's a page, let me find it. It's page 984. The nephron is really long and twisted and turned, but if you pretend it's just a straight line, these are the three kind of steps that I was just discussing with you. So I'm going to get my little writing thing here. All right, so we have all our blood flow coming in from the renal artery and branching and branching and branching. Ultimately, our last branch is before we enter into the nephron, is this cortical radiate artery and then branching into this afferent arteriole. Remember, afferent means toward and efferent means away. So we have our afferent arteriole bringing blood into this glomerulus, and we are separating by size. Small things, things that are dissolved in water, continue this way down into the renal tubule to be fine-tuned. And then things that are larger, red blood cells, your formed elements, those are going to just continue through the efferent arteriole. Now, these two guys took a separate path, but right here, you're going to see that there is the ability for things to move back and forth through this membrane. The peritubular capillaries, think about what that word means, right? So peri means surrounding. These are the capillaries that surround the tube. So they kind of weave around it. And so that's step one. So then our second step here, we have what's called tubular reabsorption. And that's where a lot of the water that went through the filter and some other things, we want to keep them. So they go back into the bloodstream. And then toward the end of filtration, there are some large things that entered the bloodstream that we really kind of need to get rid of. So we did a sort and then we fine-tuned it. And by the time we get through that third step, this filtrate that we've been playing around with has now, we now call it urine. So at this point, it's passing through this last part called the collecting duct, and eventually it's going to enter into that renal pelvis and it's going to be eliminated from the body. One example that they gave in the YouTube video, crash course video, one example they gave was emptying out your refrigerator. And so you have your refrigerator all full of stuff. You don't really know what's in it. Some of it is good, some of it's bad, some of it's, why did I buy this kind of stuff. So you go in your refrigerator and you open the doors and you look and you're like, okay, I have a big job in front of me. And so the first thing you do is you kind of take everything out. You're just like, okay, just empty this out. Let's wipe down the shelves, right? Last year, my husband, he just put stuff on top of the dirty stuff. Anyway, so you wipe out the shelves, and then you have to decide what's going to stay and what's going to go. So your initial thought is, okay, eggs got to stay, milk's got to stay, you know, whatever else you put in there that needs to stay, fresh stuff that you just bought, you put right back. And then stuff that looks old, leftovers, whatever, you throw away. But you need to fine tune from there, because stuff you put back, maybe it looked good, but it was actually expired. So you're like, oh my God, I shouldn't do that. So then you take it out and throw it away, or you put it on the counter. Then you look at the counter and you're kind of like, that's not too old. It expires in 2 days. Let me put that back. So you're fine tuning. And by the time you have everything to the end, you have your garbage and your good stuff, and you made some good decisions. That is what happens in the kidney with the nephrons. We do a sort. We gotta start somewhere, and then you sort back and forth. I give the analogy too of cleaning out your closet. First, you take everything out. You know, you decide on the stuff you think you want, and then you realize, you know what, I don't want this shirt. It doesn't fit me. It never will again. And then you look at stuff that you thought you were going to get rid of, and you're like, my grandma gave me that. Maybe I should keep it. And you put it back. That's what's happening. That's what's happening in these steps. I hope that helps. All right, let's go forward. So, looking at your glomerular filtration. I'm going to flip forward. Okay. This is slide 14 if you are if you have it printed and you're following along. So the first step is that we have this glomerular capsule, which we mentioned. This is where we're going to have our filtration. And the glomerular capsule, or Bowman's capsule, has 2 layers. It makes a cup. And so those layers are called parietal and visceral, just like in what we've been learning forever about your serous membranes. But these are simple squamous epithelium, and the visceral layer has what we call podocytes, and those are they're called foot cells. Pod or pod means foot. They're epithelial cells too, which is awesome because epithelial tissue is really regenerative and it can kind of fix itself. But these guys grab onto that glomerular capillary network, and they form these slits that act as filter pores. And this makes part of a filtration membrane. So we'll talk about that next. There's a picture of this stuff on, it looks like 985. And so let's look at that together. 983 as well. So let's look at that together. I'm trying. Okay. All right. So the podocytes or podocytes and cells of the glomerular capillaries together, they form what is called a filtration membrane. Sorry. So where I'm at with this whole thing is I see my picture, but I don't know if you guys can see it. But if you can see it, then it's blocking some of this picture. And that's why I'm looking for an actual picture for you. Some of these diagrams I've gotten from other books. And so they might not match exactly. But you do have access, well, if you're in my class, you do have access to the actual PowerPoint. And I'm trying to move my picture and I can't, so I'm sorry. Okay, so I'm going to pause this for a second again. All right, we're just going to go for it. Okay, so those podocytes and cells of the glomerular capillaries form what's called a filtration membrane. And the filtration membrane is just kind of like this, sort of, it seems complicated, but all it really is a filter, like a coffee filter, like I said. So it allows the passage or things to go through that are water and things that are dissolved in water, which we call solutes. And they enter into that capsular space, that capsule, which is right like in here, and then continue along through the renal tube. And this lies between the blood and the glomerulus. So proteins and formed elements don't continue this way. They enter back into the bloodstream. So if you look kind of closely, you can see those little fenestrations. We learned about 3 different types of capillaries, and one was known as fenestrated, and that just means it has a lot of pores in it, pretty much. So there's 3 main layers that are involved with this filtration membrane. One is the endothelium of the capillary, which is it's epithelium on the inside. That's kind of what endothelium is. And then we have slits, which are, you know, act like little, they form almost like pores. And then we have a basement membrane, which is going to be like a connective tissue type membrane. And so when things move through this filtration system, then what we have that continues through that proximal convoluted tubule, which I wrote up here, is called filtrate. Eventually this filtrate will be fine-tuned and it will be called urine. So let's look at a few more pictures. And this is what I had shown you before. This picture is on, I already gave you some pictures, or pages 985. This little candy cane looking thing is also on 985. But what you can see here, hopefully, is these fenestrations, these little pores. And then we also have these filtration slits right here. And so together, we've kind of got a really good filtration system going on. You don't need to memorize what this picture looks like, but this is what we would see with an electron microscope. This is not in your textbook. So again, hopefully you can see that there are spaces that are going to filter things by size. Some things can go through, some things cannot. Things that can go through: water, a lot of the things that are dissolved in water, and really tiny things. Water, ions, glucose, amino acids, urea. Different view, same thing. All right, here is a nice interaction, not interaction. This is showing you, remember I said you have to look at a video of this. I put the actual address on there for those of you who obviously can't click it. So that's a really good one. Nice animation, just showing you what I'm trying to explain. I'm telling you, you have to look at these animations for it to make the most sense. I can explain it, but unless you can see it conceptually, it's gonna be harder. So just do that and then come back. All right, so now we need to talk about the GFR. And if you're in my lab, this is something that we're going to be working a little bit with for your physio exercise, which is in a couple weeks. And so I'm going to give you a little overview now, and then I'll discuss it with you separately about lab. All right, so GFR, glomerular filtration rate, is really the rate at which this filtration happens. GFR is going to determine reabsorption because it determines how fast things are moving through, pretty much. If the GFR is too high, if things are filtering way too fast, then it's going to be forcing things past the beginning part of the nephron too fast, way too fast. And the body or the nephron is not going to be able to absorb things quick enough. So that's not good. If it's too low, we end up reabsorbing way too much stuff. So it's like a conveyor belt. It has to be going at the right speed for all of these things to happen in order, appropriately. And so it has to be modified and maintained like constantly. It has to be very precise. So one of the things we have is ANP. And natri, or natri, I think it's natri. That means sodium. So sodium is Na, on the periodic table. So atrial natriuretic peptide, ANP, is released from the heart. That's where that atrial comes from. It increases glomerular filtration rate. So if we have an increased blood volume, that means that we have to spill some fluid, right, in our urine. So ANP is released from the heart, and it's going to increase the GFR. It's going to make us eliminate more fluid. So if we lose sodium and water follows sodium, then we're going to reduce the volume of blood, and that is going to allow us to increase our, I should say GFR. So then blood vessels are innervated by sympathetic neurons, and we've really briefly touched upon this. At rest, we don't have a lot of sympathetic stimulation. So when we're at rest, when we're doing our parasympathetic actions, this is rest and digest. This is where you will be able, when you don't have a lot of sympathetic stimulation, you are going to rest and digest and do your housekeeping. So that's where you will eat and digest your food and absorb your nutrients. And this is where the majority of your filtrate is going to form. So your sympathetic stimulation is going to be the opposite. It's going to lead to vasoconstriction, which is going to decrease your GFR, which is going to reduce our urine output. Let me give you a page number here. Well, this is slide 20. Yeah. And so as a teacher, I don't know if this is TMI, but I can hold my pee for like ever. Part of that is because when I'm teaching and I'm walking around and I'm moving around and it's almost like it's almost like a sympathetic, I'm being very active. And you actually don't produce as much urine when you're active. When you're kind of rushing around and you've got goals to reach and you might have some adrenaline rushing. So being able to, for a teacher to go all day without going to the bathroom probably is because you're crazy busy. And maybe you do this at work. Maybe you can, hold your pee for hours and hours and hours, or you don't have to go to the bathroom at all. But as soon as you get in the car and you're a passenger and you sit back and let someone else drive, you got to pee. Well, at least I do. Sorry about that. But that's what it is. It's kind of rest and digest. So try to remember that those sympathetic neurons that are involved with, focusing on something that could possibly be dangerous, they're going to actually decrease blood flow to the kidneys, decrease blood flow to the digestive system. We don't need to worry about those right now. We need to worry about other things, muscles and blood pressure and blood sugar. That's what your nervous system is going to do, your sympathetic nervous system. All right, so that's our GFR, glomerular filtration rate. And that's on 987. 986, 987. Is this my next slide? Renal tubule, yes, okay. So what this picture is right here is more of an actual representation of what a nephron looks like. The one I showed you was just kind of like a straight line. It's actually a twisty turny weird thing with a little loop, and then at the end we have this tube. So right here, right here is our afferent arteriole going into our glomerulus. And in the glomerulus, we're going to sort things that are large don't make it through that glomerular filtration membrane, and those things end up leaving this and entering the efferent arteriole, which is going to eventually become the peritubular capillaries that surround all of this. The stuff that is dissolved in water goes through into the proximal convoluted tubule right here, and this is where our tubular reabsorption happens. And the substances that are in here that we want to keep move into the peritubular capillaries. And then we enter into this loop shape, which is called the nephron loop or loop of Henle. You have a picture on 981 that'll be really helpful here, and I'm going to show you with those capillaries in just a minute. So there's a thin part of it and a thick part of it, and then we have this distal convoluted tubule after that loop. and that is this section. So distal means further from where it started. By the time we get to the distal convoluted tubule, we've really taken care of a lot of our fine tuning. This is where our tubular secretion is going to happen, where we, put stuff back into the filtrate. Ultimately, it's going to end up in this little line here, which is the collecting duct. And going up at this picture, the lines that you see in the renal pyramids are collecting ducts. And they're going to dump into a calyx, and then into the pelvis, and then down the ureter. All right, that was our filtration. I keep, adding in the rest of the process each time. This, like I said, this topic is one that, you just have to hear it a bunch of times. I'm sorry. So the second function is tubular reabsorption. So the normal rate of glomerular filtration is pretty high. The volume of fluid, this is a crazy quote here. The volume of fluid entering the proximal convoluted tubule in half an hour is greater than all of your blood plasma. Your kidneys are insanely efficient. So once you've gone into that proximal convoluted tubule with your water and everything else, tubular reabsorption is going to happen. Proximal convoluted tubule has a big role too. So things that are absorbed during tubular reabsorption: sugar, glucose, amino acids, urea, some urea. We do have a function for urea. Not all of it though. A lot of it we have to get rid of. A bunch of ions. Things that are needed for muscle contraction, for osmotic pressure, for nervous conduction, for action potentials to happen. That all has to stay. We can't eliminate it just because it dissolved in water and went into the coffee pot. So distal stuff, further along a little bit, fine tunes that reabsorption. And so that's going to maintain all those concentrates that we discussed. Maintains appropriate concentrations of water and ions, and small proteins and peptides are reabsorbed. So when it says appropriate concentrations, that means whatever our body needs at that time. Like if we're dehydrated, we're not going to eliminate a lot of water. Our pee is going to be like super concentrated, and it's going to, how do I say this? It's going to stink. It's going to be really, really bright yellow. But your kidney doesn't care. Your kidney's like, we don't have enough water, so I don't have much to spare. If you have a lot of water, then your urine's going to be extra watery, not as concentrated. It all depends on what your body needs at that moment. All right, so this slide I got out of a lab manual at some point, but I just wanted to talk to you quickly about glucose in the urine, because I believe that's one of your physio exercises for this week or next week, or the week after. I think next week. Anyway, so let's talk about the suffix urea, which just means urine. And when you add a prefix to it, it tells you about the condition of that urine. So we have glucoseuria, which means glucose in the urine, and diabetes mellitus is the name of the disease where people can't control their blood sugar. And so when you have glucoseuria, it means that you have glucose in your urine because you have excess sugar in your urine. In your blood. So excess glucose in your blood leads to excess glucose in your urine. And so the reason that we might have high blood sugar is because, in a really nutshell, without getting into it too much, your body needs the sugar, but it needs to be, the cells need to be sort of unlocked to allow that sugar into the cell. We can think of it that way. And so with diabetes, you don't have insulin, which is involved with that. And so the cells are kind of starving, but they can't get that sugar. And so the body ends up eliminating that sugar. We have to get it out. So it's a really really easy test to see if someone might have diabetes is urine, is doing a urine test. So in glucosuria, the transporters in the proximal convoluted tubular are not able to reabsorb all the filtered glucose. And so a lot of it ends up in the urine. There's too much of it. And then we also would see excessive urination, which is polyuria. And part of that is your body's trying to get rid of this glucose, and we have excessive urination to try to do that. That can go with diabetes mellitus or diabetes insipidus, which is a different story, but we're not going to get into that. Okay, and so I know that you can't see this, but the structure or the terms here, these are your substances that you would find in urine or could find in urine, among others. And these numbers show you there are certain things we filter almost everything and return it back to the blood. And there are some things that we keep almost all of it. So your urine, as far as, here we go, erase. Okay, so as far as water is concerned, let's try again. There we go. So we filter about 180 liters of water, and we reabsorb almost 179 liters, and that means we excrete 1 to 2 liters. So we keep most of the water. Now, chloride, we filter 640 grams, and we end up getting rid of 6 grams. Glucose, we should be retaining all of it. 162 and 162. Uric acid, 8.5. We reabsorb 7.7. We do have some function for that. And creatinine, which I mentioned, is at 1.6, and we reabsorb 0 of that. And so again, I'm sorry about all this scribbly looking stuff, but technical difficulties, and I can't do anything about it right now. So anyway, you can see the effectiveness of the kidneys in this aspect. And then by testing for all of these things, you can detect the efficiency of the kidney. So we know how much is supposed to be excreted in the urine. When you do some urinalysis, you can detect a lot of this stuff in the urine, and you can sort of figure out, okay, maybe it's not functioning right if we have all these excessive numbers. And we'll talk about urinalysis a little bit later. All right, so after talking about what a nephron does, there are different kinds of nephrons. Most of them, 85% of them, are called cortical nephrons because we are mainly in the cortex. 981 has a diagram like this. I'm just looking, hold on one second. No, I see, yeah, 981 is kind of the closest thing. Okay, so cortical nephrons are mostly in the cortex, and they have all these peritubular capillaries surrounding it. It's going to, again, do that exchange. 15% of nephrons have sort of a really long nephron loop, and it dips way down into the medulla. So we call them juxtamedullary. And they start a little bit lower. So I just want my regular pointer, but I can't find it. Okay, so this guy, this is looking at the glomerular location. This is more on the cortex, because this is the cortex here. And then the juxtamedullary ones, we'd find that lower. We find it closer to this border, and so then it's going to dip a lot lower into the medulla. The main job of the juxtamedullary nephrons is urine concentration. So in other words, we're dealing with how much water we reabsorb. Okay, so So as far as blood vessels that are going to go with the nephrons, we already talked about the glomerulus being a knot of capillaries, and we also talked about the peritubular capillaries that surround those nephrons. In the juxtamedullary nephrons, the ones with a really long loop that starts a little lower down, closer to the border between the cortex and medulla, they're peritubular capillaries, but they're called vasa recta, which means straight vessels. So with a loop that long, and again, 981 has a great picture, with a loop that long, the capillaries can be straighter. Like a short loop, they're going to have to wrap around it, but in a long loop, the vessels can be a lot straighter in order to do the reabsorption and concentration of urine. So the glomeruli are going to produce that filtrate that becomes urine, and they are fed by the afferent arteriole, and they are drained by the efferent arteriole. And we already talked about how much they're going to, I mean, it's insane how much can be filtered. 1 liter of fluid every 8 minutes is craziness, and most of it is reabsorbed. So now something also that's here is that the afferent arteriole and efferent arteriole are different diameters. We talked really briefly about this when we talked about lymph nodes and lymph node flow. So the efferent arteriole is smaller than the afferent. What this means is that the afferent arteriole is larger, and blood is going to enter into the glomerular capsule area, and the fact that the efferent arteriole is smaller is going to almost allow things to pool a little bit in there and allow this filtration to happen. And again, I believe there's a PhysioX exercise that you'll be doing that goes with that. Top of my head, I'm not sure, but stay tuned for that. All right, so our peritubular capillaries are going to come from the efferent arterioles, and they're very porous and thin-walled. Porous means things will be able to pass through them quickly. Everything that enters into the urine is going to be ultimately involved with the peritubular capillaries. The vasa recta, same thing, but their job is a little bit different. They are thin-walled and they loop down into the medulla and part of the kidney's urine concentration system. Here's a diagram that's on 981. Take a few minutes to check that out. On the left is our peritubular capillary, cortical nephron, and then we have our vasa recta with our juxtamedullary nephron. So what happens in that juxtaglomerular apparatus? What happens there? At the end of the ascending limb of that loop, so we have our loop of Henle, at the end of that ascending limb, so descending loop and then ascending, there is a function that happens there in blood pressure. So the cells that are the most involved with activating something called the renin-angiotensin mechanism, which we'll get to, are called granular cells based on their appearance. So granular cells are modified smooth muscle cells, and they are going to secrete renin, which is a hormone, when blood pressure gets low in the afferent arteriole. All right, so let me give you a page number for this. I'll pause it for a sec. All right, 983 is actually the picture that you're going to see on the next slide. So it took me a while to figure that out, but it's there, 983. All right, so the renin-angiotensin mechanism we'll get to. There, at the end of the nephron loop as well, we have this macula densa. And macula densa is going to signal those granular cells when to secrete renin, which is part of that blood pressure mechanism. Part of that involves knowing kind of how watery the solution is that's passing by. excuse me. So here's our picture. On the left is just a microscopic view of the nephron. And on the right, we have our juxtaglomerular apparatus. Page 983 again. So we see our glomerulus here. We see our foot processes. We see our filtration. We see the proximal convoluted tubule, capsular space. But then right where our afferent efferent arteriole would be, we have this macula densa, which again is going to be active in that, that's going to tell those granular cells to secrete renin. All right, so now let's go into our third function, which is our tubular secretion. All right, regulation of good. Okay, so this one, the next slide is going to help with this one. But so what's happening in tubular secretion? So now we have it filtered. We reabsorbed a lot of stuff and worked on concentrating the urine and involved with water. Blood pressure stuff we'll do again in a little bit toward the end. So now we have, toward the end of our nephron, we have the distal convoluted tubule. But what's happening mostly there is called tubular secretion. That's what we're most important and most concerned with. So what happens there is that things that are in the peritubular capillaries or the blood flow, the vasa recta, all of that stuff, the things that are in there that we're going to eliminate, sometimes we need to reabsorb that. We don't always necessarily want to eliminate all of those. Some of it we do, but some of it we don't. So if we have an issue with blood pH, if that doesn't even have to be an issue, pretty much all the time, we can eliminate hydrogen ions. You know, so put these hydrogen ions from the bloodstream and put them into the tube to be eliminated. So if our blood pH does not stay at 7.35 to 7.45, if it starts to become more acidic, closer to 6, you know, lower end of the spectrum, then we need to get rid of hydrogen, because hydrogen is what makes something more acidic, the hydrogen ions. And so the secretion of hydrogen helps control blood pH. We can get rid of it, and we can also secrete other substances that we want to get out of the body or out of the blood. So normal blood plasma pH, all right, typical high protein diet. I just want to make sure you can see this because I know my picture's there. If you eat a lot of protein, then your urine is very acidic. It can be more acidic. So pretty much your urine, the pH of your urine can change drastically based on what you need your blood pH to, you know, like how many alterations have happened with your blood pH. Typically, diet that's higher, you know, a vegetarian type diet, the urine tends to be a little more basic. And again, this is because we can control that hydrogen ion concentration based on what we need for our pH regulation at that moment. So that last part says elimination of acids by cells of renal tubules secrete hydrogen into the tubular fluid. So let's look at that. This is a nice overview slide showing us what's going on in each stage. It's back a little bit. I'm pausing you. I don't have this exact picture in your lab or your lecture book, but page 993 is pretty awesome. It has a table 25.2, and it walks us through what happens in each stage. It tells you what was reabsorbed, how it happens, and it's divided, you know, into PCT, DCT, and then that the nephron loop and collecting duct. So just check it out, and you know, you don't need to memorize all of this stuff, but I do like, you know, everything that's in it, but I want you to understand what's happening in these different stages. So focus on what's happening. So the renal corpuscle, Normal GFR is up there, 105 to 125 milliliters per minute. And then when it says isotonic to blood, it just means it's the same, excuse me, solute concentration. And then we're going to do some filtering. Early distal convoluted tubule, we reabsorb a lot of water, we reabsorb salts, and then that's distal, I'm sorry. So I meant to go to proximal next. So proximal convoluted tubule, we're going to reabsorb water, sodium, potassium, all of the glucose, all of the amino acids. Looking down the list, we're also going to do a little bit of secretion, but most of our secretion happens further along of those. Loop of Henle has our reabsorption rate, percentage of reabsorption in these different spots. Our renal corpuscle, we already discussed, so we have, it kind of is weird. Okay, so renal corpuscle first, then proximal convoluted tubule and loop of Henle. And then the distal convoluted tubule is where our reabsorption happens, which I already mentioned. And sorry, here we go. Okay, and then the collecting duct, we have further reabsorption. So all we're doing, guys, is fine-tuning, 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 fine-tuning all the way through, figuring out what stays, what goes, how much water stays, how much water goes, and that's all variable, excuse me, based on what you need at that moment. I'm sorry. So, for example, if someone has excess glucose, or excess salt, we're going to see numbers a little bit different. If someone doesn't have enough water in their body, that's going to also change things a little bit. Okay. So just another view of what's happening here. All right, let's get into that hormonal regulation here. I know some of you don't have any background in hormones, so I'm going to do the best I can with explaining this without going into how hormones are created and I can't do a whole lecture on hormones right this second is what I'm saying. So I do want to explain to you that what a hormone is a chemical that is a messenger in the body. So neurons are going to send electrical signals through different aspects of the body, and then hormones do the same thing, but instead of electricity, it's a chemical, and instead of using neurons to spread this information, the hormones use the bloodstream. Okay, and so one of the hormones that we have is called angiotensin II. Angiotensin II enhances or encourages the reabsorption of sodium and chloride in the proximal convoluted tubule. Also is going to stimulate the suprarenal cortex, which is the adrenal cortex, to release aldosterone, which is also going to increase sodium reabsorption. Water follows salt. So the more salt that is reabsorbed, the more water is going to be reabsorbed. The more water you have in your bloodstream, the higher your blood pressure and volume will be. And then there's that ANP, which we already discussed. That increases your glomerular filtration rate, and that is actually going to slow down your aldosterone. And so what that's going to do is the opposite effect. So it's going to cause less sodium to be reabsorbed, which is going to cause less water to be absorbed, which is going to lower your blood volume. So angiotensin and aldosterone cause more salt to be reabsorbed, which means more water will be absorbed and blood pressure will increase. Atrial NP is going to inhibit aldosterone and speed up how fast things go through. So that's going to allow for the nephron to reabsorb less water, which is therefore going to drop your blood volume, and it's going to decrease blood pressure. So doing opposite things here. And then there's one more, and that is antidiuretic hormone. Antidiuretic hormone is going to come from the pituitary gland, and we're talking about that master gland that sits behind the eyes. A diuretic is something that makes you, it makes your urine really watery. So diuretics are things like alcohol, caffeine. So an antidiuretic hormone is going to be something that helps concentrate the urine and have it be less watery. So antidiuretic hormone also regulates water reabsorption so that we have the right balance of water in our bloodstream. So it increases water permeability of the distal convoluted tubule. This, again, the more that we can absorb, more water that we can absorb, the more concentrated the urine will be. And so if our blood volume decreases again, then we'll see a fall in blood pressure, and that's going to cause antidiuretic hormone to be released. So increased concentration of solutes in blood means there's not enough water. It means that your blood is starting to have a lot of dissolved substances in it, and that's the concentration of them increases because there's less water. And so when you have less water, your blood pressure will drop. It's just like a hose that has a leak in it. So antidiuretic hormone is released. If you don't have antidiuretic hormone, the walls of the distal convoluted tubule and the collecting ducts are not going to be able to have water pass through them very well, and we will not see reabsorption of water, and we will have diluted urine. So no antidiuretic hormone will have very diluted urine, and you're going to lose a lot of water that way. Not enough water means not enough blood volume, which can lead to blood pressure drop. So when we drink some alcohol or have a lot of coffee, then this whole system fails and we start eliminating very dilute urine. So with antidiuretic hormone, we reabsorb water and we concentrate our urine. All right, so let's say someone is hemorrhaging or they have their blood pressure is dropping. Again, we can reabsorb more water, and antidiuretic hormone is going to have control of that. It causes our blood volume to increase and help prevent shock. So I actually just talked about this. So substances that cause diuresis, which is excessive, urine flow rate, more urination, and kind of watery urine. Substances that cause diuresis slow the reabsorption of water by the kidneys. So naturally occurring diuretics I already mentioned. Caffeine and alcohol, both of those, especially alcohol, inhibit secretion of antidiuretic hormone, and that's why alcohol dehydrates you so badly. Diabetes insipidus, SIP, I always think SIP refers to drinking liquid. So antidiuretic hormone secretion in diabetes insipidus as a disorder is inadequate, or the antidiuretic hormone is working or is being released, but it's not being received. And someone with diabetes insipidus dehydrates incredibly fast, and normal urine amount is about 1 to 2 liters per day. Someone with diabetes insipidus without antidiuretic hormone being created or functioning can excrete up to 20 liters of very dilute urine a day. This person is going to be constantly thirsty, which is where I think the SIP comes in. So they're gonna be drinking, 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 and urinating, urinating, urinating. And so one of the tests that you would do for that is make someone not drink and see if they continue to urinate as much as they are. So people with diabetes insipidus, you know, they risk dehydration and blood pressure issues pretty much constantly. But with hormones, you can do a lot with synthetics. So it's not something that is incredibly life-threatening with treatment. Urinalysis is something that I was hoping we could do in lab, but it doesn't look like we're going to be able to. But it's so when we were studying blood, I tried to walk you through a little bit about how a simple blood test can tell you so much about a person's state, about their homeostasis, about infection, what kind of infection they might be fighting based on leukocytes that are there. Urinalysis is kind of the same thing. It's even less invasive, obviously, than a blood test. I mean, it's urinating in a cup and then them running an analysis on it. And so this is going to check out how much is it, what is, you know, really sometimes the odor of it. Are there microscopic things in it like casts and, you know, tiny crystals? What does it physically look like? Is it clear? Is it cloudy? Lots of things can be analyzed, and they mean, like, urine is going to tell you a lot about the state of that really homeostasis in that person. So I already mentioned a few times, normal adult output of urine is 1 to 2 liters per day, and it is 95% water. That is so variable. I don't know why they don't give a range of what percent should be water, because it's incredibly variable based on what you did that day. And then these are some of the substances that we would find in the urine, depending upon what you need in your body at the time. If there's something going on with your body, if you have an extreme diet, if you know your kidneys aren't functioning right, there are things that can show up that shouldn't be there, or there can be things that show up in numbers there shouldn't be. And you can, like I said, you can do a lot of diagnoses based on that. This is a simple dipstick test. That's what I call it. So the test stick is put in the urine, and then it's compared. That first one that you're seeing, this brown one, is glucose. And so it should be negative. Occasionally, if you eat a lot of sugar, you might see very, very low numbers of it. But this particular person has to have diabetes, because the amount of glucose in that urine is dark, and it's way, way, way high. And so, yeah, so we can do this for all of the different things that we find in our urine that we know what somewhat normal amounts should be. You can do a diagnosis based on signs and symptoms and his patient history, obviously physical, the things that you would do. And then this is going to be pretty huge. So this person, what they would do at this point is they would draw blood and check blood sugar and take the next step. Normal stuff when it comes to urine. Again, I know you can't see some of this, but the volume we talked about a million times, the color, you know, it should be a straw color is what they say, but it varies with urine concentration and diet, and the smell of it, the same thing. Sorry if this is TMI, but I had a boyfriend once who did like Atkins, like hardcore Atkins diet, like no carbs. He just pretty much ate protein and fat. Like that's all he ate. His pee, oh my God, smelled so bad. So coffee, urine has, you know, coffee makes urine smell different. So many things have an effect on urine as far as smell. So the thing here, this picture says mildly aromatic. Aromatic. Isn't aroma something good? Anyway, So the color is due to something called urochrome, which is produced from the breakdown of bile, and urobilin, which is a breakdown of hemoglobin. So these are what the colors come from. Concentrated urine is darker. Things that you eat, medications, diseases, they can all have an effect. Kidney stones can make you have blood in your urine. Urinary tract infection can have show some blood in your urine. You can see white blood cells if you have an infection. Some medications, antibiotics, some of them can make your urine red, which is crazy. Some people inherit the ability to form methyl mercaptan from digested asparagus, which gives urine a characteristic odor. That, I just gotta say, I need to take a break here from all this academia. So I guess this is still something scientific, but not everyone, when they eat asparagus, their urine smells. It's only people who genetically have the ability to make the stinky stuff from metabolizing asparagus. So there's your fact of the day, your quarantine fact of the day, something to tell your family who probably don't care, but maybe are looking for something exciting to know. So, okay, pH. Ranges in pH between 4.6 and 8, average of 6. Again, if you're someone who eats lots and lots and lots and lots of protein, expect it lower. If you're someone who is a vegetarian, expect it higher. Vegetarian diets increase alkalinity of urine because they increase alkalinity of the bloodstream, which is, you hear all about people using, alkalizing to be healthy, which is perfectly fine. The one thing that people tend to not pay attention to is that if your urine is more basic or more alkaline, it's less of a bacteria are less deterred. I hope that's the right way I'm saying this. Sorry, I think I'm getting a little loopy from being in the house. But anyway, so the higher your pH is in your urine, the more bacteria like it. They're happier with the pH of a little bit higher. So lower pH, you almost never see a urinary tract infection with a pH that low. I'm not a doctor, so I could be, stating things that are a little off course here, but one of the things that you look for when there is a urinary tract infection, you often see a higher pH, 9 or something like that. And so in conclusion, a vegetarian diet can increase the risk of having a urinary tract infection. So there's that. All right, and then the last thing here says specific gravity. Specific gravity is really, it kind of is how much dissolved substances there are in a fluid. So urine specific gravity is from 1,001 to 1,0035. 1.0 is pure water. So when you start getting up to 1.035, that's very concentrated. And closer to 1.0 would be very dilute. So someone with diabetes insipidus, we would see closer to the 1.0 because that's pure water and this is what's happening. In the morning when you wake up, expect closer to the 1.035. And when I was at a different college, my office mate, I loved her. Her name was Dr. L, and she was a vet. And she told me that, first of all, she told me that the pH of urine, if you have a dog that has a urinary tract infection, give them cranberry juice because it's acidic and it might shift the pH of urine to the acidic side. And I was like, yeah, but my dog won't drink it because he's snooty. And then she was like, well, then give it tomato juice, give it anything acidic. And I was like, that makes so much sense. But as far as specific gravity, that was another thing that she taught me, was that when vets are doing, analysis of a dog, they might ask the person to collect the dog's urine first thing in the morning because they want to see how concentrated the urine was over the night. And if it's closer to that 1.0, that could mean that the dog is in kidney failure because the urine is supposed to be concentrated throughout the day. It's supposed to be really concentrated in the morning. So anyway, let's move on. So a little bit more about what's in there. We have albumin. Your last topic I asked you, or your last quiz that you just took, I asked you as a short answer, what is albumin? Albumin is really important as far as osmotic pressure. Albumin is large. And if you see too much albumin or large things in the urine, that could mean that filtration membrane that's supposed to separate big things from small things, it could mean that it's broken. It could mean it's infected. It could mean your blood pressure is too high and it's ripping that filtration membrane in a way. Anytime you see something in the urine that really shouldn't be, try to picture again that coffee filter. So if you get coffee grounds in your coffee, it could mean that your coffee filter ripped. Or if you are filtering sand and you see seashells in the stuff that you filtered, there's probably a hole in your filter. And a lot of this stuff, this same thing can happen. If there's trauma or damage to those nephrons, if the blood pressure is too high or any of those, and we're going to see things in there that we really should not see. We already discussed glucose. For red blood cells, we'd use the word hematuria because hemato means blood and urea means urine. So the presence of hemoglobin from ruptured blood cells in the urine can occur with acute inflammation or with disease, kidney stones, tumors, all kinds of things that can, cause irritation. It also can mean that you have a bladder infection. It doesn't have to be a kidney infection. White blood cells in other parts of pus would be called pyuria. Pyo means pus. So that could mean there's an infection. Ketones, ketonuria. So ketones are going to be where your body looks to other substances besides carbohydrates, and it might turn to fats. It might look at fat and say, okay, I can't get to sugar, I can't get to carbs, I'm going to go to fats. And if that's the case, then that person might be just like on that crazy low carb diet. They might not be eating. They might be someone who has diabetes and their body can't use the carbs, so they're going for fat. So those are called ketone bodies. Bilirubin is bilirubinuria is when you have bilirubin. When red blood cells are destroyed, the globin portion of hemoglobin is split off and the heme is converted to biliverdin. Most of the biliverdin is converted to bilirubin. So you should have some, but there's a normal level. Urobilinogen is, again, involved with the breakdown of hemoglobin. It's okay to have a tiny bit, but too much could mean there's issues with the liver. Casts are little teeny tiny hardened structures. And you can see those microscopically. And then microbes, again, could be bacteria, could be yeast, sometimes protozoa, which are, little animal organisms, animal-like. And we can also see those, and we can also do a urine culture where we would see if we can grow things out on a petri dish. All right, so let's move on to just kind of the end part here is just about the structures besides the kidney and the crazy complicatedness going on there. So now we have, okay, all right, we made it. We did the filtrate, we made our urine. Now what do we do? Okay, so the nephron is going to have a collecting duct at the end, and the collecting ducts are going to run through the renal pyramids, and then the filtrate is going to be collected by a renal calyx, the major calyx, then the minor calyx, then the major calyx, and then the renal pelvis. So I'm going to try to write this. I'm going to try to use my finger this time. So the first thing, we go through the nephron. And then the second thing is we're going to go through the collecting duct. I'm just shortening it, collecting duct. And then the third thing is we're going to go and eventually empty into the renal pelvis. So there are steps in between. And I will, you know what, when I get closer to the end, you know what, I'll post that for you guys. If you're in my class, I'll post a walkthrough of all of those steps. But once we get through the renal pelvis, because this is a mess, so I'm sorry. So I'll just say it, and then we can, I'll write it out for you in a minute. Okay, so kind of going back to our first picture. So you can write this down if you want. So we have our nephron. We have steps of urine formation through the nephron. So we have blood flowing into the nephron through the afferent arteriole. There's my hand. Afferent arteriole. It's going into the glomerulus, and that's where our filtering takes place. And the filtrate moves through the tube, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. And some of those substances leave the glomerulus through the efferent arteriole, which becomes the peritubular capillaries. And that eventually will be fine-tuned and returned back to the bloodstream through the renal vein. Once the urine has gone through into the collecting duct, it's then going to run in through the renal pyramids in the medulla. And that's going to be, at the end, will be a cup called the minor calyx, which will dump into the major calyx, and that will dump into the renal pelvis, which will dump into the ureter, bladder, urethra. Now we need to talk about the ureters. The ureters are 10 to 12 inches long, thick-walled, narrow tubes. They vary in diameter. Depends on the person, depends on your age. There's two ways that urine can get down the ureter. One is peristalsis, just like you had in the digestive system. So we're talking about waves of contraction to smush it down. Also gravity. Gravity is going to have a huge role, and pressure. Because when you're laying down, you still might have to pee, right? So peristalsis is involved. The walls of the ureter have three layers. It is smooth muscle involved because, again, we don't have control over that. It's controlled by the brain, the autonomic nervous system. So we do have a mucosal lining that is made of transitional epithelium and connective tissue. So the key here is the transitional epithelium. And there are also goblet cells that are secreting mucus in that area too. So the transitional epithelium was the tissue type that could stretch, kind of. The cells overlapped, and when an organ is distended, then they can sort of unoverlap and form a sort of thinner structure, but a stretched out structure. And so we'd see that in the ureters, and the urethra, and the bladder. And then we have that middle layer of smooth muscle, and we have an outer layer of areolar connective tissue. And that's where we have nerves and blood vessels. So the ureters are going to bring that, we call it urine now, not filtrate anymore, bring that filtrate down into the bladder. So here's our urinary bladder. This is a female bladder. We know that because it's more oval shaped and because it's a very short urethra. And so when you picture what a woman's hips look like, they're a little bit wider in general, and we have this big uterus involved, and so the bladder tends to be more sort of smooshed and oblong shaped, whereas a man's pelvis is narrower and they have more of a circular bladder. And a much longer urethra because it's going to run through the penis. So what we have going on here, hang on one sec. Okay. So structures to know here. These little guys are the ureters. The ureters come in retroperitoneally. They come in behind the bladder and plug into these little spots. One right here, one at the back right here. And from here and here, this is where urine is going to build up. It's going to start draining into the bladder and it's going to fill up from the bottom. The top of the bladder is the fundus, just like the stomach has a fundus. And inside the bladder are rugae, just like we had in the stomach. Oops, I meant to erase that. Hold on. Oop. Okay, so the holes are called ureteral openings for ureter. And down here, this hole leading to the urethra is the urethral orifice or urethral opening. So we have the ureteral openings here and the urethral opening here. And when you connect these together, we have a triangle shape. And this triangle shape is called the trigon. View of the ureters. Over here we have a contrast X-ray, and you can see those ureters going down to the urinary bladder. We can see the renal pelvis. The picture on the left is just a microscopic view. Talking about the bladder now, I already mentioned male versus female. The capacity of the bladder ranges, up to closer to 1,000 milliliters. At 500 milliliters is when someone first feels the urge to urinate. It can be ignored for a little bit, but once it starts getting up higher, then the urgency increases. So when the urine volume increases, then it actually stretches the bladder and it actually rises up higher in the abdominal cavity. Again, women have a uterus, so it changes things a little bit, but then there's all these pressure receptors in the bladder as well. And then I already mentioned the trigon. And that bladder is going to be transitional epithelium, smooth muscle, again, just like the ureters. Male versus female. The next topic is going to be a reproductive system. That will be the last PowerPoint that I post this semester. Not necessarily, but the last lecture PowerPoint that I'll post. And we'll discuss the differences between male and female. But what I want you to notice in this picture is, again, not only the shape of the bladder and the location of the bladder, but I want you to notice the urethra. The male urethra is about, where's my little guy? The male urethra is about 10 inches long, and the female urethra is about 2 inches long. And so it's much easier for bacteria to work their way up the urethra in a female and enter into the bladder, versus a male's bladder is going to be a lot harder for bacteria to get up there. So men almost never get bladder infections or, urinary tract infections in general. Women are lucky enough to have that. So just looking at the bladder's histology, we have the mucosa, which we talked about being the transitional epithelium, and we do have rugae built in there. We have a muscle called the detrusor muscle, which is involved with the contraction. And then we have two sphincters at the end of the epithelium. At the end of the bladder. They are, one is more internal, one is more external. The internal urethral sphincter is involuntary. So that means that it is smooth muscle, internal, involuntary. The external sphincter is skeletal muscle. So that's the one that, when you go to pee and you hold your pee, that's the external urethral sphincter that you're controlling. This is something that, babies have to gain control over. So we do have a lot of control over it, but if there's too much urine in the bladder, then the external urethral sphincter can be like overrun, I guess you could say. And then we also have the adventitia, which is, connective tissue. We always have connective tissue not only underneath epithelial tissue, but we have it so there's always a coating on the outside of our organs. In the abdomen, we call that visceral peritoneum. If something is not in the abdomen or not in the abdominal pelvic cavity, if it is retroperitoneal, or if it is like the esophagus, it would be lined with something called adventitia. This is on the outside, which is just connective tissue. The urethra I already mentioned a little bit. Oh, I said, so a tube leading from the internal urethral orifice to the floor of the urinary bladder, in the floor of the bladder to the outside. And it's mucosal membrane, it's smooth muscle, and females, I think I said 2 inches. It's about 1.5 inches. Males, I said 10 inches. It's about 8 inches. Again, varies greatly. For males, there are different regions to the urethra, which we'll talk about in the reproductive system PowerPoint. So we don't need to do that right this second. And the external urethral orifice opens to the exterior, which we already mentioned. In females, it's between the clitoris and vaginal opening, and in males, it's going to pass through the prostate, and we'll talk about that again later. So your internal urethral sphincter is involuntary, and your external urethral sphincter is voluntary. So I'm going to show you a slide to give you an image of those sphincters. This is the male urinary bladder and urethra. Notice the shape of the bladder. We see that trigon. And then we have other structures involved there, which again, we'll talk a lot about when we get to reproductive system. But the male urethra passes through 3 main locations. And the name of the location coming immediately out of the bladder is known as the prostatic urethra, because the prostate gland is there. And then as it passes out of the body, we call it the membranous urethra. And then as it runs through the penis, we call it the spongy urethra. It's just urethra. It's just given different names in different spots. Female urinary bladder, we already mentioned a little bit. The trigon, the rugae, the ureters, and at the end, you're supposed to be able to see the urethra, sphincters. Let me give you a page so that you can look at it on your own. I'm 1003. Oh, you know what? I kind of want to go back, but I can't. So if you can go back, look at the male reproductive system slide and look for the internal and external urethral sphincters. You can kind of see them a little bit. No, you can't. So 1003. So we're almost done. We made it. High five. Wait, high five. Okay, so we have 2 slides left. One is, a couple of these I already talked about, so that's good. So the urinary tract infection is much more common in females. You know, the symptoms are going to be burning when you micturate, or micturition is urination. So there's burning, there's pain, there's pressure, there's having to pee a lot more than you did before. Yeah, mostly in women. Female dogs, female animals that have shorter urethras, same thing. So then there are kidney stones, which are known as renal calculi. And kidney stones can be absolutely massive, and I mean to the point that they can't be broken up. They have to be cut out. But they can do all kinds of things to get those stones to break up. One of the things that's pretty cool is where they use shockwave lithotripsy. So litho means stone. And they actually use like shock waves to break them up. And then you have to pass them, which means that you have to urinate them out. and is super painful. I'm sure you've all heard of or known someone who's had a kidney stone, or maybe you have, I hope not, but wicked painful. So then bladder cancer is also more common in men, and the big link there is that so in the world in general, and this is something we're seeing, okay, let's rewind. So 1 out of 3 men in China anyway, or smokes, no, I'm sorry, 2 out of 3 men in China, I'm hoping this data is correct, smoke. And so the COVID-19, obviously, that we're all in the middle of right now, in China affects men like the fatality rate is much higher in men, and that's what they attribute it to. So in general, men tend to smoke, not necessarily 2 out of 3 men like we have in other countries, but men tend to smoke more often than women. It's not a stereotype, it's just kind of a fact. And so there's a huge link between bladder cancer and smoking. So bladder cancer is more common in men. And yeah, so kidney cancer arises from epithelial cells. It's all connected very intricately. So really you can have an issue with your whole excretory system, your whole urinary system. If you have an issue in one, it can spread to others sections. As we get older, what happens? We start out with 1 million nephrons per kidney. As we get older, they just naturally stop functioning. And as we get even more older, then they don't function well. They actually shrink. We have less of them. They don't work as well. And for whatever reason, we are not given warning fast enough that we have to urinate. And so this can lead to accidents. And muscle tone is another thing. All of our muscle tone in our body, smooth muscle, skeletal muscle, whatever muscle, it loses tone as we get older. And when you're talking about the bladder, that is obviously incredibly important for your internal urethral sphincter to be functioning properly. We also have less of a desire to drink. It's loss of thirst, and so that leads to dehydration. Dehydration is not good for your body. It's not good for your kidneys. It's going to make your kidneys work harder. And then we have all of these words that can happen as you get older. So let's look at those word parts. We have polyuria it can happen based on certain conditions. So polyuria, excessive urinating. Nocturia is urinating at night. Increased frequency of urination. Dysuria means difficult uria or painful urination, or painful or labored urination. Urinary retention, so you could be holding urine. If there are stones, you could have cystic stones where there might be stones in the bladder blocking the urethra. Incontinence, so inability to hold the urine. Hematuria would be blood in the urine. And a lot of times, big things we see in men, most often things we see in men or males would be prostate enlargement. So if the prostate gland enlarges, then it can actually pinch off the prostatic urethra. And strangely, it makes you have to urinate more because it comes out slower. So it doesn't empty all the way necessarily. And then for females, we have your external urethral sphincter might leak a little bit more. Some of that is based on how many children you've had, the size of the births, the labor of the birth, you know, lots of factors. But that tends to be a women's issue later on in life more than males, but you know, it's variable. And then both people, male or female, tend to have nocturia a little more often, which is urinating at night. So that is it. Just under 2 hours. High five. You did it. Woo. Wait. Woo. That is it. So stay tuned for more videos. The reproductive one will come at some point, and I'll get them up as soon as I can. They just take hours to do, obviously. So email me with any questions, and I will talk to you soon. Stay safe, please.